Perfect. Excellent. So, well, I want to thank you everybody for joining today. It's my pleasure to, see you all participating here in our first presentation for the Neocortex seminar series. We have the privilege of listening today to the talk by Dr. Matthew Andres Moreno from the University of Michigan, who will be telling us about how has he been using the Wafer-Scale engine to power his research in evolutionary biology. And I want to remind you all that you're welcome to share your questions over chat. And Sergio, who is the co-PI of Neocortex, and me, Paula Witrago, in my role of PI of Neocortex, will be happy to help facilitate the questions. And without further ado, I'm gonna just go ahead and allow Matthew to start his presentation. Thank you, Matthew, for joining.
Excellent.
Yeah, and I would just add on the Q&A that Matthew said that maybe midway through his talk, he is going to stop to answer any questions that would have come in by then, and then there's going to be another opportunity at the end of the talk. So,
Excellent. Well, thank you for the invitation, Paula. And everybody is my screen share working okay? Okay, great. Well, then I'll just go ahead and jump into things. My name's Matthew. I'm a postdoc at the University of Michigan, and I do interdisciplinary research in computer science and evolutionary biology. To start off the presentation today, I'll kind of give a quick recap to get everybody up to speed on the domain science that I'm applying high-performance computing to, and then we'll return to talk in a little bit more detail about code actually running on hardware. One of the most exciting developments in evolutionary biology in recent decades, I think, is the increasing prevalence of experimental approaches to studying evolution. So in this kind of paradigm, rather than looking retrospectively at natural history, we create evolving systems under laboratory conditions, typically using a microbe model like E. coli or yeast, and we actually watch them evolve, and we can do experimental manipulations and really, in a hands-on, direct way, study how evolution works. A classic example of this is the long-term evolution experiment, which has been running and watching 6 lines of E. coli evolve for thousands of generations over 30 years. Experimental approaches to studying evolution go hand in hand with simulation and modeling approaches, where some experiments are not practical. They require manipulations that can't be performed on living systems, such as, for example, disabling certain types of mutations. And these simulation systems can also help to buffer against probably the hardest logistical challenge of doing evolution experiments, which is just the pure amount of time that it takes for generations to elapse, even with microbial model organisms. I went to grad school at Michigan State University, and the long-term evolution experiment was upstairs from our lab. And they had this big poster in their window about the number of generations that had elapsed in their E. coli lines. And I can tell you over my 5 years there, that number changed, but it changed very slowly. I think now they're up to 80,000 generations elapsed in their E. coli lines. And so in simulation systems, even with moderately sophisticated simulations, we can observe that number of generations very rapidly, in the course of a day or less than an hour even. However, the rate at which we can watch generations elapse in simulation systems, I think, masks kind of a side of typical single processor evolution simulations that is fundamentally smaller scale than what we see in nature, and that is population size. Typically, moderately sophisticated agent-based evolution simulations are performed with population sizes much, much smaller than we see in nature, on the order of maybe 3,600 organisms in a population. And so if you multiply out the generation rate by the population size, it actually turns out that the number of generations that we can observe elapse in a single processor simulation is about comparable to the total, or sorry, not the generations, the total number of replications that occur is about comparable to the total number of replications that occur in one flask of the Lenski long-term evolution experiment. And there's a lot of really good science and really interesting questions that we can go after working in simulations or in model systems at the scale of an individual flask. However, in evolutionary biology, there's also many questions and interesting topics that require fundamentally larger scale systems to study. One example being the evolution of rich multi-species ecologies where you have many different independently evolving populations all ecologically interacting with each other and affecting each other's course of evolution. Another example of a type of topic in evolution that's difficult to study at the single flask scale is the evolution of multicellularity. Because in this scenario, each individual in your population actually becomes composed of many kind of independent cells, and so it becomes much more costly to simulate an individual that's composed of, even dozens of cells. What I've got up on the screen is a example of a digital multicell simulation where the multicellular groups are color-coded, and they're all kind of squashed into a shared spatial arena. And by doing these types of simulations, we can get at really interesting questions about how these organisms actually select on each other, and how feedback from the environment affects the course of evolution. However, moving in this direction kind of strains what's possible in a single CPU or in kind of a multi-threading context. And so if we want to scale up to get at bigger questions, we're need a bigger boat as it was. And so I think we all have a background in high performance computing, so kind of how we scale up is pretty intuitive. But I really like kind of the way that Grace Hopper describes kind of a rational approach to scaling up computation to go after larger problems. And what she points out is that what we don't need is a bigger ox or just a single stronger computer. What we need is a team of oxen or kind of a set of computational hardware that we can divvy the problem across and that can communicate and cooperate to solve the problem together. And so typical, like classical high performance computing really is this team of oxen approach where we take a bunch of conventional CPUs and stick them together in a setting where they can communicate with high bandwidth and low latency in order to effectively cooperate on a large scale problem. However, in recent years, especially with the advent of AI workloads, there's been kind of an interesting shift in the direction of high performance computing, maybe away from this team of oxen design towards something that's a little bit more like a team of sled dogs, where special purpose hardware accelerators that bring large numbers of processors online together, such as GPUs, have become a real backbone for high performance computing. However, the capability of each of those individual processors is typically lower than you would see on, a full-fledged classical CPU. And there's with the kind of continuing development of AI, there's really exciting hardware that is coming online in this direction. I think probably the most extreme example of this kind of sled dog paradigm is the Cerebras Wafer-Scale Engine, which is a single piece of silicon with 850,000 processors stamped onto it. And so even though each processor individually isn't as powerful as a classical CPU, the kind of the size of the sled dog team opens up really, really exciting possibilities to scale up simulations. So I'll talk very briefly about kind of some of the architecture of how this particular piece of hardware that I've been working with functions. The processors on the wafer-scale engine are arranged in a grid layout where each processor...
Matt, just checking. Are you, I mean, we still see your first slide. There you go. Okay.
Oh, is that not updating?
Yeah, I mean, now it seems to be where you wanted to, but before it
Interesting.
wasn't. So, like,
Okay. Does
all
this...
the previous slides, all the
Oh,
previous stuff you said, we were just looking at the first slide, but that's fine. You'll share the slides afterwards.
yeah,
So.
I'll share the slides after. My apologies for that. Is the correct slide showing now? And this goes to the next one.
Try the next slide.
Anything, any updates?
Nope, you're still on the Cerebras
Interesting.
Wafer-Scale Engine.
Okay, then I'll just switch
Unless
to...
it's animated. I don't know.
No, no, no. I'll just switch to presenting without the full screen view because I think this is showing up correctly if I remember. See if I can at least do this. So the Cerebras Wafer-Scale Engine is a 850,000 processor chip with the subprocessors arranged in a grid topology. And it has local connectivity and communication patterns where processors can communicate with each other with low latency between neighbors on the grid. But if you want to send a message between spatially distant processors, it has to hop across the grid. Another interesting restriction of working with this hardware is actually getting data off the chip. The pinouts for device to host data transfer are on the periphery of the chip. And so if you've got something going on your simulation at the interior of the chip, to send that data out, it has to flow across the chip and then out of the edges. Another restriction of working with a wafer-scale engine is the amount of memory that's available per processor. Even though there's a large amount of total on-chip memory, when you divide it by 850,000 processors, it works out that each processor has less than 50 kilobytes of memory available. And so this, in kind of going back to the team of oxen versus dog sled metaphor, the high processor count offers really exciting potential for scaling up HPC workloads, but we need to figure out how to effectively do that within these particular hardware constraints. I'll talk next a little bit just at a high level about the strategy being used to map evolution simulations onto this wafer-scale engine. We're using kind of a pretty simple approach called a island model, where we treat each processor as hosting an independent subpopulation of the organisms that we're simulating. And so just like on an island, we might have a population of birds. On one of the processors in the wafer-scale engine, we'll have a set of genomes in the simulation, and each genome is just a fixed length array of unsigned 32-bit integers with kind of the composition and the length of that varying on what we need for the experiment. And then in order to stitch the whole population together, we use message passing between neighboring processors to enable migration to connect all those subpopulations into a united global population, just like you might see birds migrating in between islands. So let me go down here. Are my slides still advancing?
Yep.
Okay, great. So the goal for this talk will be to go over the methods and algorithm development that I've been working on to figure out how to harness this type of next generation high performance computing to this kind of emerging sled dog kind of design pattern in order to do larger scale evolution experiments in order to bring new types of questions within the scope of kind of experimental simulation-based approaches. We'll go through this in two parts. The first part being talking about kind of the core algorithms and strategy that we're using for decentralized phylogeny tracking, phylogeny being ancestry trees, which we'll go and define and introduce. And the second part, looking at how this strategy actually maps in practice onto the Wafer-Scale engine. So we'll go ahead and get started talking about phylogeny data and the strategy that we're using to collect this from our evolution simulations. So as we just went over, one of the challenges of working with simulations on the Wafer-Scale engine is actually pulling the results off the engine because of the limitations in what we can actually store on the chip and the kind of difficulty of getting data from the center of the chip out to the periphery. And one type of data that we really need to collect in order to have effective science coming out of these simulations is phylogeny data. I think probably for a lot of people, I'm guessing the last time that you saw phylogeny might have been in high school biology. But this is a kind of domain-specific type of data within the biosciences, but it really is the bread and butter of a lot of what people think about and study in evolution. Kind of as an example, there's one illustrated figure inside Charles Darwin's Origin of the Species, which of course is a phylogeny. And getting this ancestry tree data, this phylogeny data, off of the chip is important not only to kind of give us a play-by-play account of the history of evolutionary events that occurred in the simulation, but looking at the actual composition and the structure of these trees can tell us a lot about underlying evolutionary dynamics. And in applications where we're doing simulations to try to generate ground truth data for testing and developing bioinformatics pipelines, having phylogenies that come out of the simulation that we can then feed into those pipelines is really important. So I'll go over kind of the fundamentals of what a phylogeny is and how to read one. Phylogenies are oriented, typically going from the root down to the leaves. And the distance that you have to go back when you take two leaves and you trace them back on the tree and see where they connect, that distance tells you about how closely related the two organisms that you've selected are. And you can kind of think of this like the point at which these two lineages converge back in time on the phylogeny gives you the point in time when the two lineages diverged. And in this case, these species began developing independently along separate lineages. One type of data that we'll see that's kind of maybe a little bit confusing are phylogenies with tips, not just on the kind of downstream edge, but also tips inserted in the middle. And you can kind of think of this like a case where we have a fossil that's not contemporary with, the other organisms in our phylogeny, but occurred previously, but we can still hook it up and figure out where it diverged and then place it at the interior of the phylogeny as a tip. Another tricky thing about phylogenies is that there's many, many ways to draw them and orient them. And so kind of the strategy that you'll want to use to read a phylogeny is try to identify where the tips are, figure out where the root is, and then orient yourself that way. The phylogenies that we'll be discussing in this talk are organism-level asexual phylogenies. So we have our simulated organisms on the chip, and when we watch them reproduce, that corresponds to a node being added to the ancestry tree that we're tracking, kind of representing that parent to child relationship. And so as generations continue to elapse, we build up this organism level phylogeny just based on those parent-child relationships. And so in an asexual system without where every organism has exactly one parent, we get these kind of nice, well-behaved tree in the classical computer science sense structures. I'm going to skip past the bit on sexual phylogenies. And this again is kind of a very brief overview, but as an intuition for the kind of insight you can get into an evolutionary process by looking at a phylogeny, a really classical type of inference that we can do is look for really deep branches in the phylogeny to look for evidence of long-term coexistence between different sets of organisms. So in this phylogeny on the left, you can see all of the tips down here are very closely related to a single common ancestor. And so in this scenario, we can kind of infer that probably some organism in the population gained a fitness advantage and then grew and took over the population. Whereas in this scenario on the right, we have different sets of tips that are much, much deeper to go back to a common ancestor. And so looking at that branch length, we can look for evidence of kind of stabilizing ecological interactions between different groups that are causing them to persist together for longer than we would expect by chance. So the typical way of tracking phylogenies in a single processor simulation is pretty intuitive. And the kind of way that I like to think about and explain it is to kind of imagine watching a microscope slide of E. coli through a microscope, where you start off with one E. coli and then you can watch them proliferate. And as long as you can track who's who and assign all the parent-child relationships as the generations elapse, you can build up an exact record of all of the relationships and get an exact phylogeny out of the tree. And so this works great for single processor simulations where we can see the entire population all at once really easily. And however, if we scale up to a larger scale where it becomes difficult to track all of those parent-child relationships, to bring them together to build an exact tree, this type of direct tracking becomes much more difficult. And I'm happy to take questions about this, but some of the challenges that you encounter trying to scale up this direct tracking approach in a distributed system like the Cerebras Wafer-Scale Engine is the amount of memory storage and bandwidth that you need to work with all of these individual parent-child relationships. And this type of approach is also susceptible to data loss, where if you lose an individual link in your tree, you can suddenly end up with two subtrees that you don't know how they relate to each other. And so if we want to track phylogenies on the Wafer-Scale Engine, but we don't want to do direct tracking, what else can we do? How can we get this data out? And we can, of course, we can turn to biologists for inspiration, because in natural systems, in natural history, there's no, bookkeeper recording all the parent-child relationships. The way that biologists work with phylogenies is they go out and sample organisms that they're interested in, and then they take out their genetic information and do a bunch of really complicated and costly and sophisticated statistical comparisons and algorithmic work. And then kind of by looking at the structure of shared genetic traits among the organisms, it's possible to estimate the history of these lineages and when they diverge and who's more closely related to who. But working in a computational system raises, if we want to use this type of reconstruction-based approach to pull out phylogeny data without having to directly and exhaustively track it, it raises kind of an interesting question, which is a little bit backwards of how the types of problems typically encountered in work on phylogenetic algorithms, which is how should we design the genomes that we're simulating in order to maximize the efficiency and accuracy with which we can do all of these comparisons and build up an estimate of their ancestry. And it turns out, actually, this is, again, a problem that biologists have been thinking about and solved to some extent, where there's really, really interesting work going on, kind of genetic engineering of kind of CRISPR-Cas9 doodads to put into model microbial organisms, where as generations elapse in these organisms, these attachments will generate random pieces of genetic information and insert it in the genome. And then if you sample the great-great-grandchildren, it's really easy to look at what sets of organisms share which barcodes and get a pretty high confidence estimate of the underlying phylogeny. And so in order to take this approach and make it work on the Wafer-Scale engine, this requires some very careful thinking about memory efficiency, because if we're generating many, many barcodes per genome that we're simulating, very quickly, the majority of the memory that's available to us on the Wafer-Scale engine will be allocated to this tracking rather than to the underlying simulation that we're doing. And so in order to enable this type of kind of barcode approach to building phylogenies from the Wafer-Scale engine, been working on algorithms to kind of condense this type of barcode data into fixed-sized regions on the order of 64 or 96 bits, so that we can afford to kind of associate these tracking tags with each genome in our simulation. And then we can just pull these genomes off the simulation, extract their tags, and then use these tags to build a phylogeny. How are we doing on time? I think we have time to get a little bit into...
You're good? You have,
Yeah.
I don't know, 20 minutes, 25 more?
Perfect.
20 minutes if we allow for Q&A.
Then I'll get a little bit into kind of the design of how these tags are actually set up. So the general idea of this kind of barcoding tag-based approach is that each generation that elapses will randomly generate a single bit and kind of append it on to the end of our genome annotation. And so we start off with an organism with the ancestor with 1 bit, and then for its descendants, we randomly generate new bits and stick those on the end. And then we again randomly generate bits and stick those on the end. And we can continue this process as generations elapse. And eventually we'll get to the end of the run and the organisms that we want to sample. And we'll have this sequence of tracking bits that has been appended onto them as generations elapse. And we can take that sequence and do a TRIE in the sense of the tree data structure reconstruction approach, to take this sequence of bits and reconstruct a phylogeny out of it. But you're probably noticing that the challenge in this is that we can't indefinitely accumulate new bits on the end of the genome. That will require us to have, variable length memory allocations, and also, will quickly deplete our memory budget for working on the Wafer-Scale Engine. So what we do instead is make a inference quality resource use trade-off, and as generations elapse, we can actually throw away some of the tracking bits. And so we end up with kind of incomplete information where we can't exactly resolve where lineages necessarily diverge, but we can still resolve them within kind of a well-defined window. And there's really, really interesting questions in thinking about the trade-offs between which tracking bits we keep and which tracking bits we throw away. Do we want to keep kind of a set of records that's spaced evenly across the history, or do we want to kind of bias what we're keeping in order to maintain a kind of heavier coverage of more recent time? And it turns out that kind of this second recency biased strategy in many cases can give us higher quality phylogenetic reconstructions. So I think in the interest of time, I'm going to skip over going down one more level to actually talk about how we work with these data structures in practice. Basically, what we're doing is a generalization of the ring buffer. So we have a fixed length segment where we're storing these tag bits as we generate them, and rather than storing them in a left to right sequence where we would only keep the last n, what we do is use alternate write patterns to ensure that the set of marker bits in the generalized ring buffer is distributed temporally how we want in order to get high quality reconstructions. So putting this all together and kind of getting a little bit more towards the implementation, the steps that we need to build a phylogeny from genomes that we've sampled off of a wafer scale simulation is first we start off with just the raw binary data that we've taken off the Wafer-Scale Engine. Then we have to go through, extract, and decode the set of markers that we've stored in the genomes. And then we use the TRIE, tri-tree, in the sense of the data structure, prefix building approach to take these sets of lineage markers and reconstruct a phylogeny. And I've had some really great undergraduate researchers who have made contributions to different parts of this pipeline. Connor Yang has done some really great work on with generalized ring buffer algorithms, figuring out how to kind of, in a batched and vectorized way, decode and extract with high throughput markers from large numbers of genomes. Vivaan Singhvi has worked on the tree building algorithm from those markers, and then Joey Wagner over at MSU has gone through to kind of benchmark and profile this phylogeny building pipeline. And as things stand, currently we can take 1 billion genomes off of the Wafer-Scale Engine simulation, feed it in one end of the pipeline, and have a phylogeny come out the other end in about 3 or 4 hours. And so 1 billion tips is very, very large for what biologists typically work with in terms of phylogeny analyses. So this kind of the throughput of this pipeline is sufficient to kind of get us to the point where we, the reconstruction is no longer going to be the constraining factor on the types of analyses that we can do on the phylogenies coming off the Wafer-Scale Engine. We've got this sausage making machine, and now we need to figure out what to do with a billion tip phylogeny, what to do with all these sausages that are coming out of our machine. And this is not just a problem that's arising, in this in silico kind of route where we're generating these large synthetic phylogenies, but there's, really exciting bioinformatics work being done that's also generating very large phylogenies. The largest that I know of in the last few years is using single cell data to build 235 million tip phylogeny. And so another part of this project that I'm going to fast forward by a little bit is figuring out how to work with this high volume data to actually analyze these large phylogenies in a meaningful way. And one thing that we've had really good success with in this department is shifting towards a kind of data frame oriented model of serializing and deserializing phylogenies and then doing kind of statistical operations on them. And so I'm happy to talk, or answer questions for anybody who's interested if, working with large phylogenies happens to be kind of part of your side of the world. So I think I'll pause here and take questions briefly and then get into what this actually looks like running on the Wafer-Scale Cloud.
Okay, I don't see any questions in the chat. So is there maybe one live question that somebody wants to ask now? If not, maybe Matt,
We'll do it at the end. Yep. Perfect. Okay, then so we've introduced kind of this strategy for using kind of a subsample of genomes from our simulation in order to build these phylogenies and analyze, the actual evolutionary history and dynamics that are occurring in simulations. But what does this actually look like in practice? So recall that kind of the way that we have the evolution simulation set up on the Wafer-Scale Engine is as this kind of island model where each processor is its own subpopulation and we have migrations going in between neighboring populations. What we can do is as these subpopulations advance in time and go along, we can poll asynchronously using D2H memcopy commands sample genomes from each island. And so the way that we have it set up right now, we just take one sample genome from each island, and the subpopulation size is 256. So we're getting, kind of 1/256th of the whole population on the chip that is streaming off in kind of an asynchronous best effort fashion. And then once we have that kind of slice of genome data representing kind of a approximate point in history across the chip, we can just repeat that and keep asynchronously pulling out sample genomes as the chip runs and as time moves forward in the simulation. And if we kind of build this up over time, what we'll end up with is a set of, you kind of think of them like fossil genomes that are sampled from across the chip and then also across the duration of the simulation. And then we can feed those samples that we pulled off asynchronously into our phylogeny building pipeline, and we can get an approximate history of the population on the chip. And so you can see here, this is the type of phylogeny with the fossil nodes, kind of that the tips of the phylogeny aren't just at the top here, that we're also sampling organisms from across the entire history and putting them at the interior of the phylogeny. And the color coding here represents different layers. And so you can see we're sequentially pulling off layers and sticking them onto the phylogeny and building up this really detailed, approximate and incomplete, but still detailed picture of what's actually going on in our evolving population. In terms of what this looks like in a workflow, one challenge has been, given the migration of Neocortex from kind of an on-site data center that was integrated into the HPC infrastructure at Pittsburgh Supercomputing Center to the Cerebras Wafer-Scale Cloud, has been kind of the pieces of the workflow kind of around that core host device relationship, and making that all efficient in kind of this non-traditional high performance computing environment. And so what it looks like to actually do one of these jobs, I go and hop into the login node, which is a shared resource among Neocortex users accessing the Cerebras Wafer-Scale Cloud. And then I use containerized software that's provided by Cerebras on the login node to compile the code for the Wafer-Scale Engine 3 chip, and then send that off through their job submission API to a worker node, which then will run the job. It'll requisition a chip and do host device communication. Running the simulations that we've been doing to do these billion tip phylogeny reconstructions. These simulations run for about 10 minutes on the Wafer-Scale Engine to elapse 5 million generations. So we're getting about 8,000 generations a second. And then these asynchronous memcopy extractions we get about 9,000 of those elapsing over the 10-minute period. And that gives us way more than 1 billion genomes. So we throw a lot of them away and only keep kind of 1,250 of those memcopied samples. One kind of bottleneck in the pipeline is then we're done with the host device work, and we need to get all this genome data that we've generated and taken off and send that back to the login node so that it can be saved and extracted for further analysis. And kind of due to restrictions in how the API for working with these jobs is set up right now, we have to kind of chunk this data into 1 GB pieces and then stream those back one at a time. I'm generally getting about 3 chunks coming back from the worker to the login node a minute. And so it's currently the case that more time in the job is actually going to getting the data from the worker back to the login node than about 15 minutes doing that compared to actually 10 minutes running on the chip. And then we can do some kind of light post-processing on the login node, which has 128 processors available for the users, and then stream the data off of the login node back to the high performance computing center at Michigan State University, where I do the phylogeny building. I'm getting about 200 megabytes per second of exfiltration rate for the data from the Cerebras login node, and then we do the phylogeny building work at Michigan State University, and that takes about 4 hours. In terms of like a practical takeaway message that I will give to people who are setting up workflows with large data volumes on the Wafer-Scale Engine in this Cerebras Cloud context is definitely look into using rclone to get your data off of the login node and back to your home institution. I've found this tool to, like, by a magnitude of like 10 increase the bandwidth that I'm able to get data coming off the login node, and it's available as kind of through Docker Hub as a Singularity container. So you can run it on the login node without having to install anything. So definitely recommend checking that out for data intensive operations. And then the other thing to be aware of is currently needing to kind of chunk up your data and stream it back in 1 GB increments from the worker. I think I already talked about kind of some of the challenges and bottlenecks, but one thing I haven't mentioned is kind of another limitation of working in this environment is that a lot of the job aspects of the job is kind of run within a containerized environment, an SDK container that Cerebras maintains. The current version of the SDK is 1.4.0, which I think was released maybe a little bit less than a year ago. And this container currently runs Python 3.8, which is, not ancient, but old enough that it's starting to kind of become a little bit of a challenge to have all the software and runtime stuff that would be ideal for these jobs. In particular, newer versions of the Polars library don't work with Python 3.8. And so I'm hoping that in the next version of the SDK release that Python version will be bumped up to something that's not end of lifed. But now we can get into the fun part and look at pretty pictures to kind of actually see some of this data that we've been able to get off the chip and the phylogenies we've been able to build from it. So what I have on this slide is a kind of schematic of one of the phylogenies, or a subsample from one of these billion-tip phylogenies that we generated using the Wafer-Scale engine. I have in this visualization like the physical layout of the core, the CPU grid, or the processor element grid on the Wafer-Scale engine is color-coded here. So if we go kind of along the left here and then up and down, this represents kind of the actual spatial layout of processor elements on the chip, with these individual dots representing sites where we sampled a genome. And this phylogeny is just subsampled down to the canopy, so the very most recent set of genomes, with each kind of marker here in the tree corresponding to a marker for a site that we sampled a genome off of the Wafer-Scale engine chip. And you can see that we're getting pretty good spatial contiguity of in terms of closely related organisms tending to cluster together in space, which is exactly what we would expect under this type of conditions with local island-based migration. And if we kind of if we look at a little bit more data and not just kind of look at the very top of the phylogeny, but we look at all the fossil data that we've collected along the way, we can see that spatial contiguity kind of much more clearly. And so there's still some, this is preliminary data, but there's still some kind of validation work to be done, but it's promising that it seems like we're getting kind of sensical phylogenies coming off the chip. One validation work kind of piece of work that we have done is run the simulation under different conditions with kind of well-known expected effects on the structure of the phylogenetic tree that we would expect under neutral conditions versus conditions with adaptive mutations occurring. In the absence of adaptive evolution, we expect there to be a lot of diversity maintained in the evolutionary history because individual lineages aren't gaining a fitness advantage through mutation and then taking over the whole population, versus kind of looking at this phylogeny with adaptive mutations enabled, we can very clearly see kind of these repeated scenarios where we have a population, a lineage gain a beneficial mutation and take over the whole population and drive all the other lineages to extinction. And we also see kind of doing tree statistics to look at the actual composition of these trees, we can see the expected difference in the structure of the trees under these different conditions. So this again is a kind of validation in the right direction that the pipeline that we built is working as we expect. I just have a little bit more to finish up here. One of the unexpected results that we got, looking at the structure of the phylogenies a little bit more detail, was that we saw that clades of closely related organisms were kind of clustering in this top to bottom layout across the chip. So this kind of subset of closely related organisms were distributed in this vertical band across the chip. And looking at other parts of the phylogeny, we saw this pattern repeated. And if you look over time, time goes from the bottom to the top here, you can see these repeated transitions across independent lineages from blue to red. So these lineages are actually systematically moving up the chip. And these simulations were done under neutral conditions. So we did not expect to see these types of directional bias in the simulation. And if we pull out kind of individual lineages from these phylogenies, we can see very clearly many examples of these lineages kind of bouncing around on the bottom half of the chip and then migrating up across the chip. And it turns out that there was an artifact in the simulation code that we were running that was causing a migration bias. And so it was a very slight bias. I think it was like about 3% more likely to migrate up as opposed to other directions. But having this phylogeny data and looking at the spatial distributions allowed us to recognize and diagnose this kind of underlying issue in the simulation. So it's already being put to use. I'm going to kind of fast forward through the conclusion so we can get to questions. But I think there's this very interesting contrast between experimental work with evolution and biology done in vivo, tending to use sampling-based approaches. And classically, we've been able to do exact tracking for simulations to have an exact record of everything that happened in the simulation. But as we're starting to get larger scale, high performance computing resources like the Cerebras Wafer-Scale Engine with these really interesting trade-offs, it's starting to make more sense to kind of shift towards, the benefit that we can get in terms of the efficiency for accepting a little bit of data imprecision and uncertainty, as long as we still have sufficient evidence to go after the science questions we're interested in. And in parallel, kind of computer vision and benchtop techniques are starting to make perfect tracking actually possible in some constrained laboratory conditions. And so there's, I think, kind of these two sides of study are kind of crossing paths, and there's a lot that we can learn from each other. As computing hardware continues to advance, there's going to be even larger scale opportunities to run much, much larger simulations. For example, the current generation of Wafer-Scale Engine can be networked up to 2,000 chips. And so, in a future where this type of highly distributed computing with, billions of processors available, there's going to be really, really exciting science questions that we can go after. But I think we're going to have to turn to folks who work with some of the data and practical restrictions that we see, in field work in the real world for strategies to be able to efficiently exploit this type of emerging computing hardware. I'll go ahead and thank my institutional support and collaborators, in particular the undergraduate students who have contributed. This wouldn't have been possible without the Byte Boost program that Neocortex runs. That's how I got started programming on the Wafer-Scale Engine, and it's been just a really great opportunity. It's something that has helped me to develop this new line of work. And I really want to send a big thank you to the team at Cerebras and PSC for the extensive technical assistance that has gone into making this all happen. In particular, Sylvia, Layden, and Matthias over at PSC, and Paula, Mayu, Julian, and Riaz over at, or sorry, Sylvia Layden, Matthias over at Cerebras, and Mayu, Julian, and Riaz over at PSC. I really appreciate all the help. I have, it seems, an ability to generate very long email threads that are, titled like RE forward troubleshooting, blah, blah, blah. But I really appreciate all the help in making this possible. With that, maybe we have a moment or two for questions.
Thank you very much, Matthew. Questions? If nothing comes to mind now, Matthew, I think you did share your email address. And if
Yes.
it's okay with you, maybe people can just send you email, or you could also send them to neocortex@psc.edu.
Yeah, I'll go ahead and put my link to these slides because of that issue with the slides not showing at the beginning in the chat here. If it'll the chat is not loading for me, but this link will take you to the slide deck, and I'm happy to get my emails in the slide deck as well.
Okay, well, very good. Thank you all very much. Thank you, Matthew. And we will take it from here.
Thank you, everyone.